Um, Hello, I am Shaun Burd from the Kasovich group here at Stanford, and today I'll be telling you about radical pair dynamics controlled by magnetic resonance in a transgenic animal. So over the past several decades, there have been many reports of magnetic sensitivity in a wide variety of biological systems, biological phenomena. And in these examples, this is magnetic sensitivity in fruit flies and in butterfly migration. However, this field is kind of controversial, and many of these studies cannot be replicated. And possible reasons for this include the lack of sufficiently large sample sizes or sufficiently well-controlled environments where the experiments are conducted. So more recently, in this preprint, the authors tried to address some of these issues by using many thousands of samples, and they performed the experiments in a carefully designed magnetically shielded enclosure, and they can characterize the residual magnetic field down to like the nanotesla level. And they're able to observe effects in the development of African clawed toad embryos that are affected at the microtesla magnetic field level. So perhaps the most widely known example of a biological phenomena where there is magnetic field sensitivity is in avian magnetoreception. So many species of birds are known to use the Earth's magnetic field to navigate. And this has been verified and extensively replicated in many behavioral studies. In these studies, the birds are typically placed in some type of container where they cannot see outside, and they'll preferentially peck on the side of the container in a direction that depends on an applied magnetic field. Their magnetic compass can be disrupted by applying RF radiation at specific frequencies and amplitudes. And perhaps another indication of the underlying mechanism is that the birds can only use their magnetic compass in the presence of blue or green lights, but not in the presence of only yellow or red lights. So one of the theories that explains many aspects of the avian magnetoreception is known as the radical pair model that was originally proposed by Klaus Schulzen. And in this model, there are quantum correlations between unpaired electron spins in the bird's eyes. And this is associated with a protein in the bird's eyes known as cryptochrome and a flavin cofactor called FAD, flavin adenine dinucleotide. So you may wonder how a magnetic field can influence a biochemical reaction that would be required to trigger a nervous response in the bird's eye, or in fact, in any chemical reaction, considering that the KBT at room temperature is kind of many orders of magnitude away. Sorry, the energy that can be induced by a typical magnetic field is several orders of magnitude away from KBT at room temperature, or the energy of a typical chemical bond. But there are many examples of organic chemical systems, like in this photophysical reaction here, where there's a clear change in fluorescence by up to 80% in response to a small magnet. And there are many other examples of these radical pair systems that are known in chemistry. So in this work, whether or not the radical pair mechanism is involved in avian magnetoreception or not, here we show that we can engineer a radical pair system in a living animal. We use a C. elegans nematode that's been engineered to express a fluorescent protein, and we can see magnetic resonance effects at the whole animal scale, and we can even see magnetic field effects in the fluorescence at the scale of a single neuron. So you've heard a lot about the radical pair mechanism from the previous talk and others. I wanted to try and give a kind of more intuitive, simple explanation of how this can affect a chemical reaction. So here, I'll just consider a very simple system where there's an electron donor D and an acceptor A, and the donor can be optically excited to electronic singlet states, and it'll undergo what is known as an intersystem crossing to a triplet state in this scenario. So from this excited triplet state, there can be a very rapid electron transfer, and this will result in the formation of what we call a spin-correlated radical pair. Because of conservation of angular momentum, the spin states, the two electron spin states of the unpaired electrons of the radical pair will inherit the spin of the precursor. Here it's the donor in its triplet state. And because this is a two electron spin state, there are three triplets sublevels. So in general, there'll be different distributions of magnetic nuclei in the donor and acceptor molecules. And these hyperfine interactions will drive coherent singlet to triplets interconversion of the spin state of the two unpaired electrons of the radical pair. And it is this part of the process that allows weak magnetic fields to influence the outcome of this reaction. The key thing of the radical pair mechanism is that the singlets and triplet forms of the radical pair can react in different ways. So the reverse electron transfer reaction can only happen efficiently from the singlet state of the radical pair to conserve angular momentum, because it is the same angular momentum as the ground state. And this can typically happen very fast, whereas from the triplet states, this can't happen. And this can result in free radicals that can be very long-lived and take much longer to get back to the ground state. So to see how the magnetic field affects things, with no magnetic field, all the triplet sublevels are degenerates, and coherent interconversion can occur between the singlet state and all three triplet sublevels. But if a magnetic field is applied, then there's a Zeeman splitting of the triplet levels, and the altitude T plus or minus 1 sublevels become increasingly energetically isolated. And in this energy level diagram, the blue bands indicate the extent of the hyperfine interactions. So as the magnetic field strength is increased, eventually the hyperfine coupling has less and less effect on the outer triplet states, and the coherent conversion can occur only between the T0 state and the singlet state. So at low magnetic field, you would expect the reaction yield from the singlet state to be highest. And as the magnetic field is pushed up, eventually the effect will saturate, because the outer triplet levels are completely energetically isolated. So one key parameter is known as B1/2, the value of the static field that results in the singlet reaction yield going to half its maximum value. And that is can provide information about the hyperfine couplings of the radical pair. Another more interesting effect that was also spoken about in the previous talk is that if you apply radio frequency radiation, then resonant radio frequency radiation, this can recouple the triplet sublevels and allow a population that was initially in the outer triplet states to make it back to the singlet state. So when the magnetic field is moved, is close to the electron spin resonance frequency, then you expect the opposite effect to happen compared to the static magnetic field effect, and you see a resonance feature in the reaction yield. So this is known as reaction yield detected magnetic resonance because you see the outcome in the reaction products of the system. And if this singlet yield were detected using fluorescence, then this would be optically detected magnetic resonance as described in the previous talk. So going back to experimental systems, for many years, there was no well-controllable, easily engineerable protein system that displayed fairly large magnetic field effects that were easily measurable, with the possible exception of photosynthetic reaction centers. This kind of changed a few years ago when Maria Ingramo, who is here at Calico Lab, discovered that many fluorescent proteins in combination with suitable cofactors have fluorescence that depends on an applied magnetic field. This includes the ubiquitous GFP and many other fluorescent proteins. They followed up that work by showing that they could use directed evolution to engineer maglev proteins with up to, I think, 75% change in fluorescence in response to a magnetic field. And this work has been also extended by Harrison's group, as Gabby described in the previous talk, and they see radio frequency effects. And I also want to highlight that there are many conceptual similarities between the radical pair dynamics that we're using and what Peter spoke about yesterday. So for our experiments, the ones that I'll describe later, we use the red fluorescent protein M scarlet. We use this one because together with a suitable cofactor, it shows the largest magnetic field effect of any red fluorescent protein that we've measured. It can be more than 20%. So the experiment is kind of simple. You can just have a solution of M scarlet and here a photo-excited flavin cofactor, flavin photoproduct as a cofactor. And then when you apply green lights and measure fluorescence, you see that the fluorescence goes down when the magnetic field is applied. And when you move the magnets away, then the fluorescence goes back up. So I should also highlight that aspects of the mechanism for this were recently published by the Cohen group at Harvard. So to gain some more insight into what is going on, we can look at spectrally resolved fluorescence with the magnetic field off and on. And what we see is that the magnetic field effect, the fractional magnetic field effect is roughly uniform over the entire emission band of the fluorescent protein. And this suggests that what is happening is that when the magnetic field is on, there is just less protein that is able to fluoresce. And this is corroborated by absorption measurements, where when we initially, after applying green excitation to the fluorescent protein, we see the absorption maximum of M scarlet at 569 go down, and another feature at 520 nanometers grow. And then if you look at how these change in response to a magnetic field switching off and on, there's an anti-correlation between the chromophore at 569 and the species at 520. And again, this suggests that what's going on is that the magnetic field is just serving to toggle the population of the fluorescent protein in its usual state that is able to fluoresce, and another probably long-lived radical version that does not fluoresce. So for our radio frequency magnetic resonance experiments, we place our sample inside an RF resonator, and we're able to apply either transverse or parallel static magnetic fields. Again, the sample is excited using a laser, and we measure the fluorescence. So as we increase the magnetic field and measure the fluorescence, initially the fluorescence decreases. This is the static magnetic field effect, where the triplet, outer triplet levels are becoming increasingly energetically isolated. And the field at which the fluorescence reduces to roughly half of its maximum possible value is around 6 millitesla. So this is consistent with a hyperfine driven radical pair mechanism. And when the magnetic field reaches the electron spin resonance condition, you see a resonance feature form in the fluorescence, and it increases as we increase the RF field amplitude B1. We can do more experiments to characterize this resonance. We can change the resonance frequency, and we see that the field required for resonance shifts in accordance with the theory for the electron spin resonance frequency. We can also, if we use a parallel field to the radio frequency field, we don't see any significant resonance features, and this is consistent with the electron spin selection rules. The resonance line width can give us some information about the coherence time of the radical pair. From our measurements, we can bound the radical pair lifetime to around 4 nanoseconds, which is consistent with radicals in solution. So after we had measured this magnetic resonance in the M-Scarlet system together with a cofactor in vitro, we wanted to do something more interesting. So because M-Scarlet's a protein, its DNA sequence can be added into different organisms. And luckily, the Shen group on campus, it's a neuroscience group, and they use the nematode C. elegans as a model animal. They were able to help us. They grow worms that have that are genetically modified to express M-Scarlet everywhere or in specific organs or cells. And they can also generate new mutants for us using the CRISPR-Cas9 system. It's fairly straightforward. So we were able to observe the magnetic field effects and the magnetic resonance in live C. elegans nematodes. What we do is we take images of the nematodes while we're sweeping the magnetic field, and then record fluorescence. And at each point on the image, or we can divide the image up into boxes and measure the change in fluorescence resulting from the magnetic field in each one. So in contrast to the in vitro system, where we see like 20% level effects in the worms, we only see around 3% effects. And this is probably the result of the cofactor concentration that is naturally present in the cells, which we don't fully understand. We can also see the magnetic resonance at the expected magnetic field, and similarly, we can make maps of the magnetic resonance over the nematode. So most recent data is that we have C. elegans mutants that express M-Scarlet in only specific neurons, and we can see the magnetic field effect in individual neurons. And so this suggests a possible path towards magnetic modulation of neural activity. So there are many open questions. The details of the mechanism of the magnetic field effect in the M-Scarlet or other fluorescent protein systems still remain unknown, and we're trying to work them out. We also don't know the exact nature of the radical pair partner inside the C. elegans nematodes. For future applications, it would be very interesting if we could remove the requirements to have an external light source that at the moment we need to generate the radical pairs. And it would be interesting to apply directed evolution approaches to the red fluorescent protein system. So in conclusion, we've shown that radio frequency magnetic fields can influence the function of fluorescent proteins in a live animal, and that radical pair systems can be engineered in complex organisms. So in the future, probably everyone's heard of optogenetics, but unfortunately, biological tissues are often not transparent to light, and you have to implant fibers into your animal or kind of put windows in its skull, which is undesirable. It would be more desirable to use magnetic fields because they just go through biological tissues to actuate biological processes at the molecular level. And in the distant future, maybe you could think about remote control of complex organisms using the radical pair mechanism. And finally, as was kind of mentioned in the previous talk, you can use magnetic gradients to localize the fluorescence from fluorescent proteins using this system. So in this example, we generate a magnetic field null between two magnets. And because the fluorescence is maximized at low magnetic field, there'll be a bright spot, a relatively bright spot at the position of this null. And then an image can be generated by scanning this null around and imaging the fluorescence from a point detector. And you could use some lock-in detection scheme to improve your SNR. And what's interesting about this is that the image that you make will not depend on the path that the light takes out of your sample area. So it could be useful for imaging in strongly scattering media, like in like plant roots environments. And I should mention that this particular idea was originally invented by Adam Cohen at Harvard, but it's also conceptually similar to how MRI machines work. And I just want to thank everyone who's involved in this project. This is a big collaboration, in particular Nahal, who's the co-first author on our manuscript and did a lot of the work. And the group leader, Mark Hasovich. Thank you.
Oh, let's ask a question, please. Have you fused?
We've actually, Maria behind you has done this quite a lot, and I think Rebecca might speak about it later. But for example, that particular approach has not worked with M-Scarlet, I believe. Like if you have a fusion, a flavin tag, but with GFP, I think it did work. But even though the magnitude of the magnetic effect, I think, was kind of low, like the 3% level, if I'm not mistaken, or is it?
Yeah, these kinds of things are very similar to donor receptor electron transfer, energy transfer.
Yes.
Study for a long time, and varying spacer is a huge variable, and all sorts of things. A lot of literature.
Very nice work. I noticed that your Zeeman diagram shows all the energy levels are linear. Is hyperfine coupling negligible?
I guess I try to indicate the hyperfine coupling by these bands that just so like in the I guess a low field. Yeah, it shouldn't have indicated that the hyperfine coupling is linear.
Yeah, usually hyperfine coupling would curve some of the states.
Oh, yes,
Right.
Yeah, that's probably true.
Right. Thank you.
You mean the exchange interaction?
Right. So the exchange interaction will determine that gap.
Yeah.
When you're engineering a protein,
Yeah.
you want the gap to be large.
That's very interesting because that's the exchange interaction can allow you to have a magnetic field effect in much weaker magnetic fields. It like gives you like you get an inverse response.
I guess is that a limited experiment?
Oh.
To be able to support it?
Well, I think it's definitely something that we want to investigate because I know with the donor acceptor systems, you can now engineer that very well. And ultimately I think you'd want these things to respond to very weak magnetic fields. So you'd really want to investigate that and try and optimize it.
So making the radical pair larger than.
We'll take one more question for the next video. Ready? Jump in.